Okay, am I audible to all of you?
Yes,
Yes,
ma'am.
ma'am.
Okay, so today we'll be discussing the fourth uh, block of the second paper. Okay, and that deals with mostly animal biotechnology. I don't know, something is wrong with the presentation today. Okay, let me log out and log in again. Okay, some problem is there, I guess. Okay, okay. Yes. Okay, now regarding the screen, it is visible to all. The PowerPoint slides are visible to everyone.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Let's start the last unit of this paper. Okay. So this is about the animal biotechnology. So mostly we'll be dealing with topics like gene cloning, some screening of the clones means exactly how you will be identifying the clones which have your desired gene of interest. Then some applications in the form of genetic engineering, then gene therapy and tissue engineering. Okay. So if we start with basically gene cloning, I'll be telling you in very simple words. So gene cloning is basically creating or clone, we can say, creating multiple copies of something. So in gene cloning, what we do, if there is a particular gene of interest that might have some potential application or that is important from some point of view, then we try to make multiple copies of that gene. Now we know ultimately the genes will be the house of production of the protein, okay, because the gene is nothing but that segment of DNA that will actually be transcribed into your mRNA, then the mRNA will be translated into the protein. Okay, so gene cloning basically deals with making multiple copies of a gene of interest. Now, there are the certain requirements for the gene cloning or tools, you can say, that are the essential component of the gene cloning. So, one of the most vital component is the restriction endonuclease. So, the nuclease enzyme, it means the enzyme which is capable of cutting the nucleic acid that can cut both DNA and RNA. Now, then there are two types of nucleases. One is endonuclease, the other one is exonuclease. So, endo means inside. So, when a nuclease enzyme is capable of cutting the DNA or RNA in between or in the middle of the DNA segment, we call it as endonuclease. Now, exonuclease is something which can only remove the nucleotide from the terminal part. Okay, so that is what is known as a exonuclease. Now, in endonuclease, we have a very specific type of nuclease, endonuclease known as the restriction endonuclease. So, restriction endonuclease are nucleases are those enzymes which will be actually cutting the DNA at specific sequences. Okay, they will recognize a particular sequence within the DNA and they will be cutting the DNA with respect to that particular sequence. So the sequence is popularly called a palindromic sequence. So what is meant by palindromic sequence? Now if you look focus here into this sequence which is mentioned here. So palindromic sequence means the sequence from this side will be exactly the same as the sequence from the opposite side. Okay, or we can say the sequence in the both the DNA strands will be exactly opposite of each other. Now look at the sequence in the first strand, that is GGC, then G, sorry. Okay, you can see here, right? GGC, then GCC. Now, if you look into the sequence in the opposite strand, it is exactly the same. Also, GGC, GCC. So, this kind of sequences within the DNA will be known as a palindromic sequence. And usually, the restriction endonucleases, they find out a palindromic sequence like this, and they will be cutting the DNA from this particular, they means there are three different classes of restriction endonuclease, restriction endonuclease 1, 2 and 3. So depending upon at what distance they will be cutting from the palindromic or the recognition sequence, they have been categorized into three parts. Now, coming to the nomenclature, how exactly the name is given, as you can look here. So, this is the name of a restriction endonuclease, EcoRI. So, E stands for Escherichia, okay, the enzyme from which it has been derived. Then the two letters denote the species. Okay, so the E stands for the genus, the coli, from coli it has been derived, it stands for the species, and the R represents the strain of the bacteria. So the bacteria, like there are R strain, S strain, etc. there. So the R is representing the strain of the bacteria, and the Roman letter 1 represents the identification of the enzyme, means whether it was identified first, second, third, like that. So this is the nomenclature of one endonuclease. Now, if you look into, there are two different terms that might be important from short question point of view. So one is called as isocysomer, the second one is called as neocysomer. So isosysomers means the restriction enzymes that will recognize exactly the same sequence and will also cut at exactly the same position. Like CCGG, this is the recognition sequence of enzyme that is given HAPA2 and MSP1. So both of these restriction endonuclease will recognize the same palindromic sequence and they will cut it at exactly the same position. So they will be regarded as isosysomers of each other. Now, here you can see a lot of enzymes. They all will recognize the same same palindromic sequence, but the site of action will be different from each other. So those set of restriction enzymes which will actually recognize the same recognition sequence, but will act at different sites, are known as neocytomers of each other. So this is about the restriction endonucleases. You can go through them. Okay. Then coming to the other manipulative enzymes that are essential in gene cloning. Okay, so one tool we came to know that the endonuclease or the restriction endonucleases. Then there are other molecules, other enzymes, as you can observe here. These are the other enzymes like the nucleases. Okay, so already I have told you regarding the exo and the endonuclease. Then there is S1 nuclease. Actually, they have been named depending upon their means on which molecule they will be acting. For example, this S1 nuclease, it can remove the nucleotide from the 5' end of the single-stranded DNA and RNA. Okay, so that means this will not be acting upon a double-stranded nucleic acid, rather it will be acting on a single-stranded nucleic acid. That could be both DNA or RNA. Similarly, there is mung bean nuclease. So it is a single-stranded DNA and RNA specific endonuclease that removes the single-stranded overhangs from both the 3' and 5' ends. So this is another type of nuclease which will again act upon the single-stranded DNA or RNA. Then there is ribonuclease A. So this will be acting upon, so it cleaves the RNA molecule at the 3' end of the pyrimidine, that is C or U residue. So it can act only upon a RNA molecule, and specifically in which base. Now when the base is a pyrimidine, that is either cytosine or uracil. Okay. Then similarly there is ribonuclease H. So it is a non-specific endonucleolytic enzyme which is present in almost all organisms from bacteria to human. So where its specialization is what? It will act upon the hybrid. Okay, that means when there is a complex of DNA and RNA, the ribonuclease can cut or can cleave the hybrid molecule, which is a combination of both DNA and RNA. Then there is another class of enzyme called as the DNA ligase. So DNA ligases are the group of enzyme which can actually join the ends of two different DNA fragments. Okay. So they are sometimes also called as molecular glue. And also remember the endonucleases are also called as molecular scissor. So scissor means what we are using to cut. Similarly, since the endonuclease will be cutting the DNA molecule from the middle, it is called as a molecular scissor. And the DNA ligase, as it seals the ends of two different DNA fragments, it is also called as a molecular glue. Then the most important one, that is the DNA polymerase, which is highly essential for synthesis of new phosphodiester bond. So it can actually help in synthesis of a completely new DNA strand by adding the DNTPs and forming new phosphodiester bonds. And here you have to remember there is a specific fragment of the DNA during the polymerization that is called as the lino fragment. Okay, so I urge everyone to go through this and memorize what is meant by this lino fragment. Okay. Then in addition to this, there are certain DNA modifying enzymes. That means they will be actually making some changes in the DNA. So one group is like alkaline phosphatase, then there is T4 polynucleotide kinase, then terminal deoxynucleotide transferase or terminal transferase, etc. So all these enzymes have their specific functions. So you please go through them because we don't have much time to elaborately discuss this. Okay. There is a lot of things are there today to discuss. So I'm skipping this part so that you can go through it on your own. Then next we have the vector. So this is actually we can say the most crucial element of the genetic engineering or the gene cloning. Okay. So vector is, in simple terms, a vector can be known as a carrier. Okay. So this is the element that will actually carry your gene of interest. So usually the vectors are plasmids, which are extra chromosomal DNA present inside the bacterial cell. So any nuclear material cannot be acting as a vector. So the vector must have certain properties that is listed here. So what are the properties of a vector? Now a vector should have some properties that is mentioned below. First, a vector should be small in size. For convenience, the vector must be smaller in size so that it can be easily handled, and the insertion could be easier. Okay, so because it is really difficult to insert something that is huge or big in size. Then it must be self-replicating and it should be able to be purified easily. So this is also a nuclear, means this is also a nucleic acid, but it is different from the genomic DNA. Okay, so this is the plasmid DNA. So it should be easily, means we should be able to differentiate it from the chromosomal DNA easily. Then it should be easily introduced to the host cell. The host cell, which will actually take the gene of your interest, must be able to accept this vector. So it should have unique multi-cloning restriction enzyme sites for cloning insert DNA. So this vector must consist of specific sites where you can actually put your gene of interest. So those sites will be nothing but your restriction enzyme sites, where the restriction enzyme can go and cut and take out the part of the vector DNA, and in place of the vector DNA it can insert your gene of interest. Okay. Then it should have suitable marker gene, which is very essential to find out whether the actual insertion of your gene has taken place or not. So this is actually useful for selection of the means vectors, like selection of the transform vector, whether the vector has actually taken up the gene or not. Then it should be easily maintained inside the host cell. That means it should not interfere with the physiology of the host cell. It should not be disintegrating. So this should actually remain stable within the host cell. And based on the function performed inside the host, the vectors can be broadly categorized as cloning vectors and expression vectors. So the vectors are broadly of two different types. The cloning vector that will only carry the gene of interest and will multiply within the host cell to just increase the number of the copy of the gene. But the expression vectors, what they will do, and they will actually express the protein which should be encoded by the gene. So these are the example and the capacity of different type of vectors. The most common vector, as I had discussed, is your plasmid vectors. Okay, but the demerit is what? The plasmid vectors, they can take up only very small fragments of DNA. Okay, like only up to 15 kilobase pair. Now there are phage vectors that are derived from the bacteriophages. Then cosmid vector, the plasmid having the cos site of the bacteriophage lambda. Then there is BAC, that is bacterial artificial chromosome, which are nothing but the fertility plasmid of the E. coli. Then there is yeast artificial chromosome or YAC, which is derived from the Saccharomyces cerevisiae. Okay, so these are actually, as you can observe, the intake capacity of this BAC and YAC are comparatively much higher than the rest of the vectors. Okay. So depending upon the size of gene they can take up, they have various applications. Now this is an example of a vector, plasmid vector actually, PBR322, which is very commonly used. So like you can see, this vector has all the properties we have discussed, and most importantly, it is having a origin of replication. That means it is capable of self-replication. It has multiple restriction enzyme sites. You can observe a numerous restriction sites are there. So these sites can be used for cutting and taking out the DNA, the vector DNA, and in this place your gene of interest can be inserted, so that when this vector will go into a host cell, there it can increase the copy number of the desired gene. Similarly, this is another most frequently used vector known as the M13 vector. Okay, so this is the genome of the M13. Means this is actually a bacteria. Okay, so this is the structure of the M13. Sorry, not bacteria. This is a phage, means bacteriophage. Okay, so this is the M13 filamentous phage, but it has a means wide genome. That the size of the genome, you can see there is 6.4 kilobase pair. Okay, so this M13 genome is also frequently used as a vector to for cloning purpose. Then this is a cosmid vector. So this is similar to your plasmid vector. The only difference is that it is having a cos site. Okay, so this cos site is nothing but a specific gene which is a part of the bacteriophage lambda. So then comes the bacterial artificial chromosome. So as we discussed, they have high intake capacity. That means within the bacterial artificial chromosome, you can insert a gene which has almost 300 kilobase pair sequence. Okay, that means the ATCG, whatever sequence is there in the DNA, like that 300 and kilobase pair means more three zeros you can add. Okay, that means you can put a gene sequence that has almost 3 lakh base pairs in this bacterial artificial chromosome. And these are the sites, that is this is the origin of replication. Okay, so this is the cause site might be present here, and these are the restriction sites which are present. And this is a chloramphenicol resistance gene for selection of the transgene, means transgenic vector. So there are various other sites which are essential for maintenance of the vector, multiplication and maintenance of the vector. Then similarly, this is an yeast artificial chromosome, and as you have seen, this is capable of taking up DNA of very large size. I think 1,000 base pair, 1,000 kilo base pair, something like that. Okay. So when you need to insert very large size genes, you can use the East artificial chromosome. Then comes so these are all actually your cloning vectors. So cloning vector means they will simply carry the gene. Once inserted into the host cell, they will simply multiply in number and they will increase the copy number of the gene. But there are certain vectors that are called as the expression vectors. So what they do, now in addition to carrying the gene, they will also express the gene within the host cell. Like they are having this promoter operator sequences which are highly essential for expression of the gene. So they will not only carry the gene of your interest, but they will also produce the protein which is supposed to be coded by the gene. So that is why they are called as the expression vector. Okay. Like for example, if I say we are inserting a insulin gene within here. Okay. So what these cloning vectors will do, they will simply carry the insulin gene inside the E. coli bacteria and they will keep on increasing the copy number of the gene. Maybe the vector is creating 1,000 copies of itself. So now the E. coli is having 1,000 copies of the insulin gene, but the cloning vectors will not express the insulin protein. Okay, but if this is an expression vector, or if you insert the gene in an expression vector, when the copy number will increase, at the same time, the insulin protein will be actually synthesized inside the host cell. So that is the utility of the expression vector. Then comes the second part, that is the gene cloning 2, which is actually the construction of the DNA library. So what we have in a library, so in a library we have a collection of books, right? Similarly, the DNA library means that is a collection of the genome of an individual organism. So there are different types of DNA library. One is called as the genomic library. So as the name suggests, the entire genome will be the entire genome will be fragmented and it will be put inside different vectors, and all the vectors will be collected together to construct the genomic library of an individual. Like let's say for one organism such as C. elegans. So the entire genome of the C. elegans will be fragmented and they will be put inside different vectors, and all the vectors will be collectively stored in one place, which we will call as the genomic library. So these are the steps of forming a genomic library. The cells with the genomic DNA. Okay, so then the cells are ruptured by lysing and they are treated with some proteinase, means to degrade the proteins, and the genomic DNA is isolated. Then the genomic DNA, as you can observe, this is a large DNA sequence, okay, or a large DNA molecule. So it cannot be stored easily. That is why it is made or it is broken into smaller fragments. So it might have multiple restriction sites where the restriction enzymes will cut and will create means small fragments like this, okay, by restriction digestion. Then each fragment is actually put into the plasma, the cloning vectors that are mostly BAC or YAC, as they can carry large fragments of gene, and then these means vectors that carry the genes are actually transformed into the bacterial cells. Now the bacterial cells will be carrying different fragments of this particular organism. So when you are searching for the gene, any particular gene of interest, you can look for the genes within the set or clone of the transformed bacterial cells. So that is how the genomic library is constructed. Then there is another way of constructing the genomic DNA library by using the lambda phage. So as you can observe here, this is actually somehow a larger segment of the DNA is taken up. The high molecular weight eukaryotic DNA, the DNA is definitely more than 100 kilo base pair. Means almost the DNA size is containing 1 lakh base pairs. Okay. So you can cleave the mixture with H3 and ALU1 enzyme, very partial digestion. Okay, and the size fragments are usually around 20 kilo base pair. So EcoRI methylase to block the EcoRI site so that the EcoRI cannot digest this DNA. Then the blunt end ligation with the EcoRI linker molecule. And then you can subject it for the digestion by the EcoRI. But since this part is already protected by your methylation, the EcoRI cannot digest this inside part, but it will be acting upon the terminal parts to first form sticky ends like this. Okay. Similarly, you what you can do, these parts you can secure with a parallelly constructed size fragment to means this here what is done, the alpha chadon the 4a DNA, that is the replacement vector is used. Then you can anneal the cohesive ends of the lambda phase DNA, and you can subject it to the digestion by the EcoRI restriction enzyme. So you will have sticky ends like this. Okay. Now these internal fragments can be means if you observe here, this part is actually removed. Okay. So this part is removed and you are having the internal fragments like this. Then you can size fractionate to remove the internal fragments, and whatever sticky ends will be available. No, sorry, this is actually creating multiple fragments like this. So you need actually this one. Okay, and these small fragments are not needed. So by subjecting it to the means you can use any kind of gel analysis where the you can isolate these vectors, or you can isolate these fragments based on their size. So these are small size which will be actually moving towards the end of the gel, so you can just cut and remove them, and you can isolate this larger fragment. Okay, then simply you have to join these two sticky fragments to form this kind of genomic library. Okay, and these segments are again reintroduced into the bacteriophage lambda where they will be secure. So this is the way where you can construct a large DNA library, or you can construct library that is consisting of DNA bases of around 1 lakh base pairs. So like this you can have multiple means here you have 20 kilo base pair. So in one segment you are having 40 kilo base pair DNA. Like that the rest of the DNA you can store in this different fragments, and you can pack them inside the bacteriophage particle. Then coming to the screening of genomic library means when you are using for example, if you look here, okay, so you are putting these vectors into the bacterial cell, but it is not necessary that every bacterial cell will take up your desired vector. So how you will find out whether the bacterial cell that is that you are analyzing or that you are considering has your gene of interest or not? So that is what is called as the screening of the bacterial DNA. Sorry, the screening of the genomic library. So one way of screening or isolating or finding out whether it is containing your desired vector or not, screening by the probe hybridization. Okay, so probe hybridization, how it can be done. If you look into the means the procedure, what is happening here. So we can start the transformant colonies growing on the agar surface. Okay, so you have already introduced the vector, and now you want to find out which of these bacterial colony has actually your desired vector. So you can retain a master plate, means you can make a replica of this plate, and you can use another plate, or you can what we can say subject the other plate for analysis. Okay, so what is done, the replica plate onto the nitrocellulose disc is placed on the agar in the petri plate incubate. Then you can see this is the petri plate where the bacterial colonies are growing. Okay, so what you can do, you can just put a nitrocellulose disc on this plate. So once you put the nitrocellulose disc, what will happen? This bacterial colonies will be transferred from this plate to the nitrocellulose disc. So once the nitrocellulose disc is removed, it has already taken up your bacterial colonies from the petri dish. Then you have to follow multiple steps, like you have to lyse the bacteria with 0.5 NaOH. Then you have to neutralize it, and you have to digest the proteins using the proteinase enzyme. Then you again wash it and bake it at 80 degrees centigrade. Now you have got the DNA print. Since you have already used the proteinase, most of the proteins will be degraded. And now what you have on the nitrocellulose paper, that is only the DNA print. Then you can hybridize it with the P32 label probe. That means the DNA has the phosphate within it, right? The phosphate groups are there, or they are essential part of the DNA. So what you can do, you can use a probe. Probe means a DNA segment that contains a radiolabeled phosphate P32. So this probe can go and hybridize with the existing DNA segments. So wherever the hybridization will take place, so this probe is actually having a complementary sequence to the vector you have introduced in the transformed bacteria. So this probe will go and bind with the desired vector that is present. So wherever the sequence will be available, only there the probe will be binding. So what you can observe now, wherever you are getting this autoradiograph, means that is only containing the radiolabeled phosphate. So that is the colony having your having your desired DNA. So you have to compare it with the master plate you have written. Okay. So out of these 5 colonies, bacterial colonies, only this colony has the vector that you have introduced it. So that is how you will be screening, or you will be selecting which bacteria is actually transformed. Then you can also do the same by PCR amplification. Means you already know the sequence of the genes that you have cloned into the vector. So you can design a primer complementary to that DNA sequence, and simply you can subject it to the PCR. So if there is amplification, or you can observe the band after the PCR is done, that means the gene of interest is present there, which could be amplified by the primer you have designed. And if the vector will not contain the desired set of DNA, then there will be no band after the PCR is done. So from on that basis also, you can find out which of the colonies have taken up your DNA and which have not. So these are the ways by which you can screen which bacterial colony has your vector of interest, okay, or which has actually taken up the cloning vector. So this was the construction of the genomic library. So genome means the entire set of genes that is present within an organism. Or we can say the entire DNA that is present in an organism is stored in the form of library. Now there is another library which is called as the cDNA library, that is the complementary DNA library. So what is happening here? Now the cDNA is constructed actually, means the DNA, it is actually consisting of, as we know, the exon, intron, everything will be there, but every part of the DNA is not actually transcribed into mRNA. But in cDNA, what is done, the part of the DNA which is actually transcribed into mRNA, only those parts will be conserved in the library. So what is done in construction of the cDNA library? Now first, from the cell, the mRNA is extracted. Then the complementary DNA is synthesized. Means the mRNA will have a particular sequence. So in using that as a template, a complementary DNA is synthesized. So this is the first strand and the second strand synthesis of the cDNA. Then from the using the mRNA as a template, the first strand of the complementary DNA will be synthesized. Like for example, the mRNA is having a strand like this: A U G C U A, like this. So what will be the complementary DNA to this? Now this with the DNA will have T A C G, then A, then T. Okay, now this is a template again. So the second strand of the cDNA will be synthesized like this. from against the T, there will be complementary A, then against the A, there will be complementary T, then against C, there will be complementary G, then against G, there will be C, then T and A. So that is how you again got a double stranded DNA, right? From one strand of the mRNA, first you form the first strand of the complementary DNA. Then again from the first strand of the complementary DNA, you have got the second strand of the complementary DNA. And this DNA will be further cloned or will be amplified and will be stored. So this is what is called as a cDNA library, complementary DNA library. So this is the way by which you can isolate the mRNA. So we have already discussed that the mRNA will be having a poly A tail, right? So in the 3' end, they will be having a polyadenyl tail, which is called as a poly A tail. So we are using oligo DT beads. So these beads, they have multiple T's. So when you will pass or you will load the RNA sample, the mRNA that has multiple A's will get attached to the beads, okay, and then you can purify them further or you can elute them further and you can isolate the mRNAs that is present in the RNA sample, because the RNA sample might also contain tRNA, rRNA, but only the mRNA will have the poly A tail. So by using the oligo DT beads, you are only purifying the messenger RNA, okay. So there are various ways by which you can construct the mRNA. You can prepare the cDNA. So one method is called as a self-priming method. So what is done in the self-priming method? Now this is the mRNA, and in the mRNA, a oligo DT primer is used because mRNA will have multiple A's at the 3' end. So in complementary to that, you are using a oligo DT primer. So the T's will go and pair against the A's. Now you can incubate it with an enzyme which is called the reverse transcriptase. Okay, so the reverse transcriptase can add deoxynucleotides against the mRNA and form a copy of the DNA. So now after this, there will be, let's say there is some other sequence like C, G, A, T. Okay, so whatever might be the sequence, now the reverse transcriptase can keep on adding nucleotides as per that. Okay, and the strand will extend in 5' to 3' direction. So when the C DNA strand is completed, the RNA strand is hydrolyzed. So hydrolyzed means you have to separate them. Okay, so you have separated the mRNA or hydrolyzed, or you have removed. Now what you are left with is cDNA. So now the cDNA can use itself as a primer to synthesize the other strand. So incubate it with the DNA polymerase to synthesize the second strand of the DNA. Okay. Then you can use the S1 endonuclease cut loop. Okay. So and you will be having the double stranded DNA. Now again you can incubate it with the terminal transferase to add single stranded tails like this. So the double stranded complementary DNA is ready for insertion into appropriate cloning vectors. Okay. Then there is another method by which you can also construct the cDNA, that is called as the homopolymer tailing. So this is also somehow similar but slightly different. So what you are having here, so again like the poly A tail, you are using a terminal transferase plus DCTP, means cytosine. Okay, so you are, in addition to this, you are just adding the cytosine bases in the 3' end of both the strands. Then you can simply degrade the RNA, and once the RNA is degraded, you can anneal it with a oligo DG. Okay, means in complementary to the cytosines, you can add the guanines, and once the guanines are added, the further elongation can be done by using of DNA polymerase. So in this way also you have got the complementary DNA, double stranded complementary DNA. So one you can do by the self-priming method by using a oligo DT primer in the beginning, or you can simply add terminal transferase plus deoxy cytosine triphosphate, which are actually nothing but the cytosine residues which will be added to the DNA strand. Then comes the screening of the clones, or exactly how you will identify which bacterial colony has taken up your gene of interest or not. So there are various ways by which you can identify or you can screen them. So one is by the DNA hybridization using a specific probe. So probe is nothing but a known sequence of DNA. Okay, so probe is a known sequence of DNA which has complementary sequence against your target DNA. Okay. So usually the DNA probe is labeled with a fluorescent dye for easy detection because it will have a distinct color which can be easily visualized. So what is done? The target DNA sequences and both the probe, they are denatured. Denatured means from the double stranded structure is broken and they are made into single stranded. Now since they have separated, if the probe is having a complementary sequence against the target DNA, it will go and bind there. Okay. And after binding, if the binding is successful, that means if the target DNA has complementary base pair to your probe, then definitely you can observe some kind of fluorescence emission, which can be detected by a system, and you can find out whether the gene sequence or the gene of your interest is present in the target DNA sequence or not, because you have designed your probe accordingly. Okay. Then it can also be done by Southern blotting. So Southern blotting is actually a DNA blot technique. So there are three types of blottings you should keep in mind. One is Southern blotting. Blotting is related to DNA. Then you have Northern blotting related to RNA, and you have Western blotting related to protein. So since this is Southern blotting means you will be actually constructing a DNA blot. So what is done here? As you see, there is a genomic DNA. You have made multiple fragments by digesting it with the restriction enzyme. Then you have subjected these fragments to agar gel electrophoresis. So what is the utility of this? Now here, this will separate the DNA strands based on their size. The small size fragments will move to at the other end, okay, like this, and gradually you will have a gradient of fragments from low to high molecular weight. Then you just put the gel on a nitrocellulose or nylon filter. So by capillary action, whatever thing is present in the DNA gel or agar gel will simply move to the nitrocellulose filter. Okay, like for example, you just imagine you have a gel and you put a pepper behind that. So whatever is there in the gel will simply leak to the pepper. So similar thing is happening here. And again, you can hybridize it by using a phosphate 32 radio labeling probe. So wherever there is complementary sequence is there, because the probe will have a definite sequence, and that sequence, if it is complementary to your target sequence, it will go and bind there. Okay, and then you can expose it to the X-ray film, and you can get the auto radiograph, and you can know exactly which segment is having your gene of interest. Okay, so that is how you can find out whether your desired gene is present in a particular segment or not. Then just as I was telling you, there is Northern blotting technique. So this is exactly the whatever thing I discussed, how they are actually being transferred from the gel to the cellulose, that is given here. So Northern blotting technique is used to monitor the gene expression. The sample RNA from the target organism is isolated and separated using the electrophoresis on a formamide agar gel, followed by transferring the isolated RNA to a support membrane. Usually the support membrane is a nitrocellulose membrane. So in the presence of a specific buffer, this can be accomplished using the capillary transfer technique. The membrane is next either backed at a high temperature or subjected to UV cross-linking, which causes covalent bonding of the RNA to the membrane and prevents the nucleic acid from being washed away during the later processing. So if you see, look here, the same thing is happening. Whatever is there on the gel, it has been transferred to the membrane. Okay, similarly, since here the DNA has been transferred, because we have put the DNA in the gel here, in that case, in the northern blotting, instead of DNA, you have used RNA. Okay. So these are the steps of the northern blotting. Same thing, the RNA molecule is run on a denaturing agar gel. The gel electrophoresis separates the fragments. Staining with the ethidium bromide allows the visualization of the RNA molecule. The blotting transfers the RNA bands to a nitrocellulose filter. The gel and the filter allow the transfer buffer to pass through and reach the paper towel. This results in a nitrocellulose filter with the RNA fragments precisely transformed from the gel. So a probe for a particular gene that has been radioactively labeled is exposed to the filter. In case of the transcript is produced, the probe will bind to the RNA. And the filter is exposed to X-ray film for autoradiography. A band on the film identifies the fragments containing the desired gene. So here you can look in the picture. Okay. So this is the RNA that has run on the denaturing gel. So denaturing gel means it will cleave or it will cut the RNA into smaller fragments. Okay, and then you are transferring whatever on the gel is there to the blotting paper. So the blotting paper is nothing but a nitrocellulose membrane. Then the RNA is transferred to a RNA binding membrane, the nitrocellulose membrane, whatever it is. Then you have to hybridize these RNA fragments with your desired probe. Okay, and then you just expose it to the X-ray where the autoradiogram will be developed like this. So as you can observe here, this particular segment or the fragment of RNA which is around 5 kilobase pair is containing your gene of interest, because that is the location where your hybridized probe or the radio labeled probe has gone and hybridized. Okay, the rest of the fragments does not contain your gene of interest. Only the segment of DNA that is, or the fragment of RNA which is around 5 kilobase pair in size, contains the gene of your interest. Okay. Then comes the colony hybridization. Okay, so this is also similar to whatever we saw in the southern blotting. So you people are getting it or not? Because these are techniques. So I don't know how many of you have been exposed to the laboratory condition. Anyone in your BSc have good exposure to laboratory wet lab experiments like isolation of DNA, RNA, etc. Has anyone done? Because these things actually little bit difficult to understand theoretically, but once you do it experimentally, it becomes easier to grasp. So somehow, even if you don't have any exposure, you can go through different videos that are available on YouTube. There you will actually get to know how the experiments are done and how exactly the results are coming out and analysis is being done. Okay, because I do understand that theoretically it is not very easy to grasp and analyze exactly what is happening without a practical exposure, because these are techniques, right? So techniques means something which you have to do. So I recommend all of you to go through the whatever techniques we are discussing today in to the means lot of videos are available. Laboratory based experimental videos are available in YouTube. So I recommend all of you to go through that and just make sure that how this technique is actually being performed. So these are whatever we are studying. These are just the procedures. Okay. So in what step by step you will be conducting this, so in the video you will get to know exactly, like have you ever seen a agar gel, any of you, and you have done a DNA in that agar gel?
No, ma'am.
uh, okay, so I request all of you to means go through the visual representation that will actually whatever I am discussing. No, you can correlate that time. Okay, so that is how exactly it is done. See, all the molecular biology experiments, they are very precise. Means you will be actually dealing with some microliter, nanoliter quantity. Okay, so the okay, just let me tell you. Just 1 second. I don't know why. Okay, can you all see the video now?
Yes, ma'am.
Okay.
Yes, ma'am.
So just have a look how the
This video will
electrophoresis is
take you through the process for loading and running
being done.
DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber.
Okay, that is how a gel is looking like. Okay, you can see. And this is the
Add
buffer
electrophoresis
that
running
is
buffer to the
being added.
reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a
This
DNA
is called a
sample, slowly
micropipette.
depress the
Okay.
plunger on your
In
adjustable
which you will be
micropipette
taking
to the
out
first or soft stop.
the DNA sample.
Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button.
The DNA is mixed with the
When loading the
sequence here.
samples, keep
Okay.
the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well.
The
Slowly
different
apply pressure
samples
to
are
the plunger
loaded. Like
button
you
and
can see
observe as
the
the
wells
sample
are
fills the
marked
well.
like the
Make
1,
certain you
2,
stop at the
3,
first
4,
stop on the
5 like this.
pipette,
Okay.
pause, then
So you
slowly
can add
remove the pipette
like
while
around
keeping your
8
thumb down on the
samples
plunger.
at a go.
Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run,
Okay,
the
so
dye
this
front
is
moves
how
from the negative
whatever we are
electrode
talking
towards
about
the
from they
positive
are
electrode.
migrating. So the smaller will be the DNA fragment. So there you can see two bands are there. Actually, one band is actually the dye. Okay, so the dye will be actually moving before the DNA sample, and the DNA sample will be just behind. Okay. So So whatever autoradiogram you are observing here, right? So these are nothing but the pictures of the gel that you just saw. Now you might be correlating a little bit. So you go through every technique that we will be discussing today so that it will be easier for you to understand. So I am just explaining the theoretical part, and for the practical exposure, you just visit the YouTube videos. Okay, so there are quite good explanations are there. So you will get to know. So then the next is what we are discussing is colony hybridization. Okay, so what you will get here. Uh, it is similar to something that we have already discussed in the hybridization of the selection of the clones. Okay, so here also same thing is done. You have a master plate. Okay, so in the master plate, you can prepare a replica and you can put a nylon membrane on the replica plate. So place the membrane on top of the agar containing the separated bacterial colonies. Okay. Then invert the membrane and lay over the new agar surface and allow the colony to regenerate on top of the filter. So this is the replica filter on the new agar. So you have put a membrane here, you have taken off the bacterial colonies from here on the membrane and you are putting the membrane on a separate agar plate where the new colonies will grow. Now you can remove the membrane with the attached colonies and soak in the successive solutions. So what are the solutions? First you have to denature, then you have to neutralize, wash and dry and fix the DNA. Now the bacterial DNA are fixed on the membrane. And again you can follow the steps like hybridize with the label probe, then wash the probe, means if the probe is not bound, that should be removed, and then you subject it towards the autoradiography. Now whichever colony has the gene of your interest, only there the probe will go and bind. So when you will subject it to the X-ray filming, what you will get? Only one colony will show the positive value. That means only this colony of bacteria has the gene of your interest. That is why it was able to bind with the probe that you have designed and you have sent, labeled with a P32, and you are getting a positive result for this. The rest of the colonies, they do not have the desired DNA or the desired gene of your interest, so they are not forming any kind of hybridization with that of the probe. So no autoradiograph is seen for them. So you have to identify the positive colony and you can put it in the liquid culture medium for further provocation. Okay, so here you will take it and you will put it in the liquid culture medium where this colony will further keep on replicating and your gene of interest will be multiplied, let's say 1,000 folds. Then similarly, there is also plaque hybridization. So when creating gene banks or libraries, the phage vectors are frequently employed instead of plasmids. So this method is similar to colony hybridization, but the generated plaques can be hybridized to check for the presence of the particular DNA sequence. So plaque hybridization has some advantages over the colony hybridization, mainly due to the smaller and more precise area of the filter to which the DNA is bonded. This allows the screening of more plaques during a single hybridization. So on a standard Petri dish, the distance, for instance, thousands of plaques can be screened during a single hybridization. So the number can reach hundreds of thousands when utilizing the larger containers such as baking dishes or trays, and additionally multiple replicates can be obtained from the sample plate because the technique does not alter the placement of plaques and does not smear them the way colonies can. So this implies that many probes can be used to test the same batch plaques for hybridization. Now what is actually plaque? If you look into this, is let's say bacteria. Okay, so the bacteria can be infected by a bacteriophage, and the bacteriophage can go and lyse the bacteria. Okay, so this lysed bacteria is actually forms a clear zone in the petri dish. If this is the petri dish, this is a bacterial colony, but if this bacterial colony is infected with the bacteriophage, the bacteria will be killed by the bacteriophage and you can see a clear area on the petri dish. So that is what is called as a plaque. So if you are using the phage vectors, okay, so the phage vectors means what? They will be carrying the DNA of the bacteriophage, and the bacteriophage DNA you are using as a vector. So, if the phage vector is having your gene of interest and it is used against or it is infected to a bacteria, it will go and lyse the bacteria and you can form the plaque. So, whatever phage has your gene of interest, that phage will go and form a plaque like this, and you can, instead of looking for bacteria colonies, you will be searching for the plaques where the phage has infected the bacteria. So that is what is called as plaque hybridization. Then coming to some very important techniques. See, we have been assigned only 4 classes, okay? So I cannot do anything about that. I have to finish it by today only. I have been assigned 4 classes for all the 4 blocks, okay? So I don't, I mean, I can't do anything about that. Okay, then comes the immunoscreening, means there are different techniques which are used in disease diagnosis. So 3 major techniques we will be discussing. One is the ELISA or enzyme linked immunosorbent assay, then RIA radioimmunoassay, then chemoilluminance immunoassay. So in ELISA also there are again 4 different types: direct, indirect, sandwich, and competitive. So let me explain the principles of these one by one. Okay. So first one, as you can observe, is the direct ELISA. So in direct ELISA, what is done? So all the principles of immunoassay are based on the fact that the antigen and the antibody, they interact with each other, okay, and they form a antigen antibody complex. Okay. So here, what is done in direct ELISA? Usually, the antigen is coated into a microtiter plate, and then it is washed for any unbound antigen that might be present. Then an enzyme, or sorry, then an antibody linked with an enzyme is put in the well. Now the antigen is actually complementary to the existing antigen. Sorry, the antibody is now complementary to the existing antigen. So if there is specificity, then the antibody will go and bind with the antigen. Okay. Now this antigen is this antibody is having a enzyme linked to this. So that is why enzyme linked immunosorbent assay. Now this enzyme is capable of converting a chromogenic substrate into a colored product. So now after the antigen and antibody has been bound, has taken place, you will be using a substrate. So the substrate is actually converted to the product by this enzyme, and this will give a color to the plate. So depending upon how much color you are getting, you can predict the concentration of the antigen. Okay. So this kind of ELISA is called as your direct ELISA. So for example, what is this? If I want to say, like you want to find out whether a person is infected with HIV or not. So HIV is your human immunovirus. So the virus has certain part like protein or something that is actually the pathogenic substance. So you have isolated the pathogenic substance, or that is the antigen, and you are using a antibody which can bind to this HIV antigen to find out the concentration of the HIV antigen. So if the HIV antigen percentage is higher in the sample, so what you will get? you will get a very intense color here, because more the antigen, more antibody will go and bind to that. More antibody binding means more number of enzymes are available, and more enzymes mean more conversion of the substrate into the color product. So high intensity of color. So that is the use of the immunoassays. Okay, you want to find out the concentration of a antigen in an unknown sample. Then what is done in indirect ELISA? It is little bit different from the direct ELISA. In direct ELISA, only one complementary antibody bound to enzyme is used. This is also called as a primary antibody. But here what you can observe is, there is the antigen is coated on the wall, then it is washed for any unbound antigen that is present, then the antibody which is complementary to the antigen is given. So if there is a specificity, then the antibody will go and bind to the antigen. Now again you have to wash it to find out if there any unbound antigen is present there. Now you are using a second antibody. So the second antibody is linked to the enzyme. So that is called as a secondary antibody. So this secondary antibody is actually complementary to the primary antibody. So wherever there will be complementary means binding will be there, then only if you are using the chromogenic substance, then there will be a conversion of the color or change in the color that you can see, like from blue to yellow it is happening, right? Then you will be measuring the intensity of the color to find out what could be the possible concentration of the antigen in your sample. Then there is another type of ELISA called as the sandwich ELISA. So in the sandwich ELISA, as the name indicates, the antigen is actually sandwiched between two different antibodies. Okay, so this is the first in direct ELISA we used to first add the antigen to the wall, but here in sandwich ELISA first you will add the antibody to the wall. Okay, then you will be so here you want to find out the concentration of the antibody in the serum or in the sample. So you have first used the antibody to coat the wall, and then you will wash to find to remove any kind of unwanted antibody here. Then you will be using the antigen against it. Like for example, you want to find out whether a person is infected with hepatitis B. Okay, so if I am infected with hepatitis B, my blood serum will be having the antibodies against the hepatitis V virus. Okay, so but I means the doctor does not know what is the concentration or at what level I am being infected with the disease. So what he or she will do, they will be extracting the antibody from my serum and they will put it in the well. And then we know if we are suspecting a hepatitis virus, so we will be using the hepatitis antigen here. Okay, so if the hepatitis B antibody is there in my blood serum, the hepatitis antigen will go and bind to the antibody. Okay, now you will use a secondary antibody linked to the enzyme that has the ability to convert the substrate to a chromogenic product. So you will put the substrate. If the secondary antibody binding has taken place, then definitely a color will change. And depending upon the concentration of the antibody, there will be high intensity color. If the concentration will be less, the color will be less, or the intensity of the color will be less. If the color is high, means many antigens, many antibodies are there to which many number of antigens can bind, and more the number of antigens binding, more will be the binding of the secondary antibody and more will be the product formation and higher will be the color. Okay. And since here the antigen is sandwiched between two antibodies, this is called as a sandwich ELISA. Then the last type is a competitive ELISA. Okay. So competitive ELISA means what? Here it is slightly different from the rest of the ELISAs. So here we are using two different types of antibody. So we are using an antibody, or we are incubating an antibody with an antigen that to be measured. So what you will do in the well, you will mix the antigen antibody mixture to the already antigen coated wall. Now these are actually the radiolabel antibodies. Okay, sorry, these are the radio level antigens that you have already used on your antibody, and you are using this against the on level antigen that is present in the wall. Now, again, then what you will do, you will be using this conjugate secondary antibody, and then you will add the substrate and you will be measuring the color. So here what is happening, this is the sample hour, this is our sample. Okay, so what we can observe, if here the sample will be more, that means if the number of antigen in the wall will be more, then what will happen, that these whatever radio level antibodies you are using, they will be replaced by these antigens. Okay, means this antibody will now release the radio level antibody antigens, and you will they will go and bind to the existing antigen in the well. So more is the number of antigens here, or higher is the number of antigens in the well. Okay, so low will be the radioactivity. So there is a inverse relationship. Up to this, there was a direct relationship, means higher is the concentration of antigen or antibody, more will be the chromogenic substance. But in competitive ELISA, as there is a competition between the label and the on-label antigen, so what will happen, if if the concentration of the on-label antigen will be more, this means chromogenicity will be less, and vice versa. So that is why this is called as a competitive ELISA. Okay. Then this is the principle, actually how exactly the ELISA is working. So the what is done basically, first there is the coating. Simply the control antigen is observed onto the well in ELISA plate in the coating buffer. Then you can remove the liquid and wash the plate. Then you have to block the antigen so that it does not move away. So a buffer containing unrelated protein is used to block the free sites on the wall, so that no more antigen can be bound to the same wall. So within one well, only one antigen should be present. So then you can remove the liquid and wash the plate. Now prepare the sample, that means the sample with which contains unknown antigen, and you can detect the antibody mix. Then load the sample. Okay, so add the test sample and mix to the wells. Then remove the liquid and wash the plate. Then again you can use the detection antibody, add the enzyme conjugated secondary antibody, and remove the liquid and wash the plate. Then read out. The substrate is catalyzed by the enzyme to generate a colored product that is called as a chromogenic substance. So in this example, the antigen concentration in the sample was low. The antibodies bound the control antigen that had been observed to the plate in the coating state. So if there will be low, then the color change will be less. Then comes the radioimmunoassay, or RIA, usually called as. So this is also somehow similar to your sandwich ELISA, competitive ELISA. So here also you are using two types of antigens. Okay, so one is the radioactive antigen or radio level antigen that is already bound to your antibody, the first antibody, and these are the unlabeled antigens, or these are the sample antigens actually. So now again here also there will be competition between the radio label and the on-label antigen. So if the concentration of on-label antigen will be more, they will keep on replacing the radio label antigen. Okay, so they will precipitate the antigen antibody complex with the anti-immunoglobulin, that is the secondary antibody, and you can measure the radioactivity of the supernatant in the supernatant for the free antigen. So that means if you in your sample, like for example, if we are screening for same, let's say the example of hepatitis B, okay, so you have used a antibody against the hepatitis B virus, and you have used the hepatitis antigen by radio labeling it. Okay, Now, if in your sample the concentration of hepatitis B is very high, so what they will do? Now the hepatitis antigen of the sample will replace your radiolabel antigens. Okay, and now this primary antigen, which is containing your on-label antigens, is bound to the secondary antibody. So that means what? More radiolabel antigens are now free, so they will have more radioactivity. In contrast to this, if the sample, the viral load or the antigen load, if in your sample will be low, then what they will do? They cannot replace these radiolabel antigens on your antibody. So most of the radiolabel antibody will still remain bound to the antibody, and the radioactivity will be very less here. Okay. So on that basis, you can predict whether your sample is containing more or less amount of suspected antigen, or whether what is the load. Okay, whether they are high load or low load, that can also be predicted. Okay, any questions or any queries from anyone? If you go through the text, no, you can correlate. If you think this is difficult, no, nothing is difficult over here. If you read it mindfully, then nothing is difficult. Okay, so they are quite easy also. Now coming to the next type of the immunoassay, that is your chemiluminescence assay. So here it is like all the assays, they are more or less similar only. So here also similarly you have an antigen and you are using a complementary antibody or primary antibody. Then you are using a secondary antibody and you are using a enzyme, that is HRP, which stands for horse-radish peroxidase. Okay. So horse-radish peroxidase and alkaline phosphatase, these are the enzymes that are frequently used in this ELISA. No, this diagram is not in the book. I have taken it from out some sources. Okay, I have cited the source here also. And the other previous diagram, I think there is a book on immunology called QB. If you can find it, that is the standard reference book to follow any kind of immunological study. So this RIA picture is from QB. Any edition you can take. I have taken it from the 5th edition, and this I have taken from a paper I have cited here. Okay, the reference of the paper I have given here. So this is also similar to whatever we have discussed till now. This is the chemiluminescence signal. This is the substrate or substance which will be catalyzed by this horse-radish peroxidase to give a colored substance. Okay, and this intensity of the color intensity is measured, and based upon the intensity, we can predict the concentration of the antigen or the antibody, whatever we are analyzing. Then coming to the next, that is the characterization or mostly by the sequencing, how to find out the sequence of a particular DNA segment. So one of the most common method that is used is the chain termination method by Sanger. So this is called as the Sanger sequencing. Now what they do in this technique, so as you can observe here, the ingredient for synthesis of new DNA strand are usually DNTPs. Okay, so N could be A, T, C, G, anything. So this is called deoxy adenosine triphosphate or guanosine triphosphate, then thymidine triphosphate and cytosine triphosphate. But if there another oxygen is removed, then it becomes dideoxy. Okay, so as you can observe here, DDNTP. So this template DNA, when this primary will form complementary DNA strand against this template, it should be adding the DNTPs. But if instead of DNTPs, one of these DDNTPs will be present here, then it cannot be elongated any further. So in this particular reaction mixture, what is done, alongside the primer, this kind of DDNTPs are added. Okay, so what happens here? For example, here if it is A, okay. So against A, what should be added here? T. So if against A, T is added, and after A, let's say there is C. Then this after this T, a G will be added, and the chain can elongate. But here, what problem can occur is because of the presence of these DDNTPs against, if here is an A, okay, and instead of T, if this dideoxy TTP is added here, so what will happen? No further elongation will occur. Okay, so as you can observe here, like this, whatever sequences are there, each sequence will be blocked by a presence of these dideoxy NTPs or DDNTPs. So that is how you will be getting this primer extended into multiple fragments against the template of multiple length. And when you will analyze it, you will get the sequence of the DNA that is present on this template. Okay, so this is what is called as the Sanger sequencing or the chain termination method, because here we, after adding a DDNTP, we are just blocking the extension of the chain. So once you will if you put all the strands together, what you will get, let's say here it is C, here or DC. Okay, dideoxy cytosine is there. Then here it is, after C there will be G. Like this, you can, if you combine them all, you will get the entire sequence of this particular template DNA. So that is what is called as chain termination method or the Sanger sequencing method. This is a very preliminary method to find out the sequence of the DNA. Then in today's era, there is the next generation sequencing method is there, which is pretty advanced. And here, as you can see, a very short fragment can be sequenced, right? Like you can observe, the length of the DNA is not very long. This is a short fragment. But in case of next gen sequencing, multiple adapter molecules are used, and simultaneously you can sequence a huge amount of DNA. Okay, so essentially all fields of biotechnology have been revolutionized by NGS, which has also become a critical method in genomics. It allows the scientists and doctors to examine the biological system at a degree and resolution precisely unthinkable, thanks to the excellent throughput, scalability, and the speed of data collection. So this is basically a high throughput technique where you can generate a huge amount of sequencing data by very precise methods. Okay, so before the invention of the next gen sequencing, the Sanger sequencing using the dideoxy chain termination method was the prevalent method for determining the DNA sequence for more than 30 years. Then it was regarded as the gold standard for discussing the genomic differences. As the genome-wide sequencing is now made possible by high throughput NGS, the genomic research has recently tended to use DNA sequencing as a critical molecular research tool. Anyways, the principle is not being detailed here. If you wish to know, you can refer to any kind of video that explains the principle of next gen sequencing. Okay. Then coming to the next part, that is genetic engineering. See, I have no choice of over that. I have been assigned only one class, and in that class only I have to finish. I don't have any other choice. The link that has been given to me, I can use it only for 4 days. Okay. So just I am trying my best to convey the principles to you. Rest you go through, you go through the materials. I think the material is available online. You should have gone through the material before coming to the class, so that it would have been easier for you to grasp. Anyone who had means studied coming before to the class, at least in this particular topic? That is why the material has been made available to you long back ago, right? See, these like as per my knowledge, the distance programs, they are not like regular classroom teachings. It's more kind of self learning. You should be learning, and wherever you have problem, we are here to sort that problem out. So I means it is expected that you must have gone through the material previously, and wherever or what are important topics are there, that the counselor will try to explain to you. And if you have any doubt or queries regarding any particular concept, that should be clarified here. Okay. So that is how, in my view, the distance thing works. And regarding any problem you are having, you should contact them. Okay, I don't have any control over these things. Whatever I have been assigned, I have to complete things in that time period only. Okay, so I'm really sorry regarding that. Okay, so let me continue whatever things we have left. So genetically modified microorganisms, basically they are engineered. So the cloning and expression of the eukaryotic genes in the prokaryotic system, that is one of the vital applications of the genetic engineering. So what are the steps involved in this process? The obtaining and cloning the eukaryotic gene to be expressed in the bacteria. So what gene you want to express, first you have to isolate that or identify and isolate them. Then you should increase the copy number by cloning, and then you need to use, or you need to introduce it into a host bacterium. So that leads to the transformation of the bacterial cells with the cloned gene. Then selection of the cells having the desired genes, that we have already seen how you should be screening it. Then induction of the bacterial cells to express the inserted gene to derive the translated protein. So preferably the expression vector is used here. Whatever vector can carry the copy as well as express the protein that is present on the gene. Then collection and purification of the eukaryotic protein. So the first thing that was used for this purpose was the insulin. Okay, so you all must be aware of the insulin, that is the enzyme which is highly responsible for keeping our blood sugar level in a controlled way. So people with impaired insulin function, they have various health issues, problems, starting with the diabetes, and it might lead to many other problems or dysfunctions in the body. So in order to meet, so there are a group of people who are unable to produce sufficient amount of insulin. So that is your diabetes 1 type, those who are, so they cannot produce sufficient amount of insulin. So they are dependent upon an external source of insulin. So now from where such a huge amount of insulin will come? So what we can do, we can isolate the human insulin gene, and if we can introduce it successfully to a bacterial cell like E. coli, and E. coli can have multiple copies, like 1,000 to lakhs number of copies of the bacterial cell. So if it is introduced into expression vector that can actually produce insulin inside a E. coli bacterium, then we can have sufficient amount of insulin that can be used for injection purpose. Okay, so this is the structure of the insulin. So usually insulin is synthesized as proinsulin, and then the disulfide bonds, whatever are there, they are actually cleaved, and you get the mature peptide. Okay, so this is the way by which the insulin is being expressed. So this is the insulin gene A subunit. This is the promoter. This is the lag Z gene, and this is the antibiotic resistance gene of the expression vector. Then you are transforming it into the E. coli. So we are having two different subunits, like one the gene A subunit, the other one is the gene B subunit. So the beta gal or insulin A fusion protein is accumulated in the cell. Similarly, since the lag Z is there, so the beta gal and the insulin B fusion protein is accumulated in the cell. Then extract and purify the beta gal and insulin fusion proteins and treat the fusion protein with the cyanogen bromide to cleave the A and B chains. Okay, then purify and mix the A and B chains to form the functional insulin by forming the disulfide bond. So that is how, by inserting the insulin gene into an expression vector, you are transforming a bacteria, and you are increasing the number of the bacterial cell by multiplication, so that you can have a large amount of both insulin A and B chain, and then you can purify them by various methods are there to purify the proteins. Then you just combine both the subunits through the disulfide bond to form the active insulin, which can be directly used. So this is the industrial way of synthesizing insulin. Then next comes, there is another growth hormone that can be also produced in a genetically modified bacteria. Okay, so similar to the vectors, like it will be having different type of sequences, like the origin of replication is there, the lag promoter and the lag synthesizing gene is there, that is the lactose of from the lactose operon. Then ampicillin resistance gene is there to find out or screen the selection of bacterial cell that have taken up the gene of your interest, and this area contains your HGS, that is the human growth hormone coding sequence. So the structure of the first gene construct and the vector used to produce the HGH in the E. coli. Okay, so these are the vector sites, and this is the DNA fragment that is actually containing the coding region of the human growth hormone. Okay. So this is, look at it carefully. This is the cDNA, because only the exon or the coding part has been converted into the DNA form. The gene is not used directly. Rather, from the gene, the mature mRNA has been produced or isolated, and the mRNA has been converted into DNA and has been put into the vector. Okay. So this is the native growth hormone, the prolactin receptor binding site, the growth hormone receptor binding site, and this is the modified growth hormone. Okay, so altered prolactin receptor binding site, this is the growth hormone receptor binding site. So this is the diagrammatic representation of native HGH and modified HGH with altered prolactin receptor binding site. The altered prolactin receptor binding site does not allow binding of the HGH with the prolactin receptors and avoids unwanted effects of the prolactin. Okay. So if you observe, if this is the growth hormone, okay, in the native growth hormone, two types of hormones can act on this. Like prolactin can also act, growth hormone can also act. But sometimes in a cell we do not need the action of the prolactin. So there the problem will arise, because the natural growth hormone is having a binding site for both. So in that case, we can construct a modified version where only our desired growth hormone will go and bind and interact. And this site has been mutated. Okay. So as per our requirement, we have modified the hormone. The same thing that is explained how exactly it will be behaving inside the cell. So this is the peptide linker. This is the growth hormone. This is the receptor, growth hormone receptor. And within the cell membrane, this is the cell membrane, this is the growth hormone receptor, and it can form a dimer like this. Okay, so that is a reversible reaction. So wherever growth hormone is required, this dimer breaks down and the monomer is being recognized by the growth hormone receptor. And this is the structure, just for your information it is given. Then comes the transgenic plants. So first of all, just understand what is meant by transgenic organism. So any organism having a different gene. So trans means actually other. Okay. So any organism that has gene from some other organism will be regarded as a transgenic organism. So it might be an animal, it might be a plant, anything. So basically the transgenic plant, they have genes from other different plants. So let's understand the basic principle of generating a transgenic plant. So usually there is a bacterium called as Agrobacterium tumefaciens, which is used in construction of the transgenic plant. So this bacteria contains a very specialized plasmid called as the TI plasmid. So TI stands for tumor inducing plasmid. Okay. So these are the various regions of the TI plasmid. As specifically you can observe, it has the genes that can code for auxin and cytokinin, which are the growth hormones of the plant. Okay. So this plasmid, to whatever plant cell it infects, it can cause the tumor. Okay, so it can multiply on its own. So how exactly the transgenic plant is created? This is the Agrobacterium tumefaciens. Okay. So this is the TI plasmid. This is the this is actually the genomic DNA of the bacteria, and this is the plasmid of our interest. So this TI plasmid, it also contains different sites that can be digested by the restriction endonuclease. So the tDNA, this is the virulence region. Virulence region means which is actually responsible for causing the disease in the plant. Okay. So what is done? This particular region is cut by the restriction enzyme. Okay, the restriction cleavage site, and in this site, your gene of interest is actually inserted. So insertion of the gene into the plasmid using the restriction enzyme and the DNA ligase. So this is the how exactly the gene of your interest is created. So what is done? Now in the from the plant cell, the inserted DNA carrying the foreign gene is actually taken out. Then you can culture the plant cells, and those plants will be grown into the plantlets, and then you will have the transgenic plant. So here, sorry, I came to that part. Here actually what is happening, you have, this is the gene of your interest, or this is the plant DNA, and this is the gene of your interest. So this is also cut by the same restriction enzyme, and it will have exactly the complementary side binding sites against the tDNA. Okay, so they will go and integrate here, and you will have, this is the your vector that is carrying the gene of your interest, and there is the promoter for the targeted gene expression. Now this is what is called as a recombinant vector, or this is a recombinant TI plasmid, and this is the antibiotic resistance for selection whether that gene of your interest has been inserted into this or not. Okay. So now this TI plasmid is inserted into the plant cell, and now the plant is grown, is having the gene of your interest. So you can also go for gel electrophoresis of the gene of your interest, like that is chitinase. So chitinase means the enzyme that can degrade the chitin. Okay. So this is how a transgenic plant is made. So what are the basic steps? You are using the TI plasmid as a vector, and you are cleaving the TI plasmid by using the restriction digestion enzyme, and you are also forming a segment of your gene of interest by cutting it with the same restriction enzyme. Okay. Then you are joining them by the DNA ligase, and you are just now you have already created the recombinant plasmid. The recombinant plasmid is introduced into the plant cell. The plant cell is grown in the culture medium, and the each means culture will grow into a mini plantlet, and when they are grown up, the plant will be having, in each of the cells of the plant, this desired gene of your interest will be present. So whatever gene of interest you have inserted, the plant will now be expressing that particular protein. So we will see some of the examples, like exactly how it is useful. So here the generation of a herbicide resistant plant. So herbicide means a chemical that can kill a herb. Okay. So this is the viral promoter, and this is the EPSP synthase, means this is actually the enzyme which is acting against the herbicide, that is the chemical. Okay. And this is the termination signal. Similarly, what you will do in the TI plasmid, you will be inserting the fuse gene. Okay, so this is the promoter is highly essential. Without the promoter, this particular EPSP synthase will not be expressed in the TI plasmid. So you have fusion gene inserted into the TI plasmid. The recombinant plasmid is inserted into the means R radiobacter, that is the host bacterium for growing the plasmid. Then the R radiovector transforms the plasmid carrying the EPSP fusion gene into the plant chromosome, resulting in high levels of EPSP synthase, making the cell resistant to glyphosate nucleus. Okay, sorry, glyphosate nucleus. The cells from the plant leaf, the calluses with the EPSP fusion gene grown in the medium containing the glyphosate. Now, since this particular gene, that is EPSP synthase, can degrade the effect of the glyphosate, so this plant will not die in presence of this particular compound. So the plants regenerated from the calluses are glyphosate resistant. Okay, so that is the utility of the transgenic plant. You can introduce any property or any character that you want to be present in your plant through the TI plasmid. I think everyone must have heard about the BT toxin plants. Okay, so the BT actually it comes from Bacillus thuringiensis. So the cotton cells, so they are actually highly infected by a certain type of pest. Pest means a insect that usually destroy the cotton plants. Okay, so a BT gene is used to protect the plant against the pest. So how exactly it is done? The promoter of the BT gene turns on the production of the BT toxin, the protoxin in the plant tissue. So the protoxin is converted into the when the insect will be eating this particular plant tissue, inside the gut of the insect, the enzymes will activate, will cleave the protoxin into toxin. Now the toxin, what it will do, it will simply create holes in the gut or in the stomach of the pest. The gut cell will rupture and the pest or the insect will die. So that is how the plant will be protected against the insect. Then there is another application of the transgenic plant, that is the production of the golden rice. So golden rice is actually a variety of rice which is highly rich in vitamin A. Okay. So there is a precursor molecule of vitamin A that is called as the beta carotene. So this beta carotene is actually synthesized from a precursor molecule called GGPP, that is geranyl pyrophosphate. So there is a gene called as phytoin synthase, phytoin synthase, that is PSI. So this PSI gene converts this GGPP to phytoin, and the bacterial CRT gene converts, it is actually stands for carotene gene. Okay, so it converts the phytoin into lycopene. Then the lycopene beta cyclase, that is the lysi gene, is converting the lycopene to beta carotene, and finally the beta carotene is converted into vitamin A by the endogenous human gene. So this particular transgene having this particular transgenic plant is having multiple transgenes induced into it. So they all are taking part in the same metabolic pathway to produce vitamin A from GGPP, the precursor GGPP, so that the rice is actually enriched with a high amount of vitamin A. Now the transgenic plants can also, there are many other applications also, you can go through in your study material. Then another very important application is the phytoremediation. So phytoremediation means using the plants against the environmental pollution. Okay, so the plant phyto term is related to plant. So phytoremediation is the use of plants to remove, destroy or isolate hazardous substances and pollutants from the environment. So transgenic plants overexpressing the phytogelatin synthase have been designed and experimented to remove contaminating metals and organic compounds from the environment. Like let's say there is a mining area or something which is heavily contaminated by heavy metals like lead, mercury, etc. Okay. So now you have designed a plant. That plant contains the gene called as phytogelatin synthase. So what this particular enzyme can do, now when the plant will take up this heavy metal, the enzyme can fix these compounds into its leaves or somewhere, and when that plant is killed, now that environment is free from this pollutant. or sometimes the plant can also release them into environment in other forms as byproduct like gases or something. So that is how this polluted environment is is cleaned by use of transgenic plants. Then there are transgenic animals also. So similar to plants, they are also animals which will be containing foreign genes or genes from other animals. So the procedure for or steps for production of transgenic animals include construction and cloning of the transgene, obtaining the oocytes, fertilized ova and embryos, then integrating the transgene into the oocyte, zygote or embryo, then culturing and transferring the zygote or embryo to the surrogate mother, creation and production of the homozygous transgenic line. So these are the steps of producing the transgenic animal. So I think here there is the production of transgenic animals. Okay, so how it is done? And now you have isolated the somatic cells from one animal and you have cultured it. Then you have you can do whatever modifications you want in this somatic cell. Okay, then you can take up egg cell from some other animal. So the egg cell is usually the enucleated, means the nucleus is removed because you don't want these genes to be expressed. Rather, you want the genes that you have modified to be expressed in the future cells. So these two cells are fused, and now the fused cell is developed up to the blastocyst stage. Okay, so you know how the development occurs. The means the morula, then the blastula, then the gastrula stage comes. So they are usually grown up to 16 cell blastula cell or blastocyst stage. Then the blastocyst is implanted in the surrogate or in the foster mother. So this is the foster mother or the surrogate mother. So whatever child is born, the child will be born with these genetic qualities, whatever you have modified. So this is now a transgenic animal. So transgenic seed was created by the nuclear transfer. So these are some of the means applications of the transgenic animal, like the transgenic mice can be used for studying different type of human diseases. Like here, this is the mouse model to study the Alzheimer's disease. So Alzheimer's is a neurodegenerative disease. So as you can observe, these are the neuron cells. Okay, the healthy neuron cells, these are normal. Now sometimes what happens, there is formation of amyloid plaques like this. So there is a beta amyloid protein that are produced. Okay, so usually there is a enzyme responsible which can remove this beta amyloid when they are produced. But if that enzyme is defective because of some reason, usually it occurs due to old age. Okay, so the amyloid will not be removed and they will keep on the number will keep on increasing and they will start accumulating and they will form plaques like this. Okay, so they will affect the normal functioning of the neurons. So it leads to loss of memory, language disturbance. A lot of problem can happen. So this is actually the procedure of formation of the amyloid plaque. So these are the beta amyloid protein precursor. So because of the faulty proteolytic cleavage, there is production of the beta amyloid protein, that is AB40 or A beta 42. So inefficient clearance of AB40 and AB42. So usually when they are produced, they should be cleared. But if they are not cleared, they will keep on accumulating and this will lead to development of the dense senile plaques at the end of the inflamed nerves. Then coming to the transgenic mouse model to study the cell death. So similarly, the transgenic mouse are created to study the functioning of particular gene, how they will be acting. Like here is an example of a genetically modified mice that is explaining or that is expressing a defect in the how exactly the diphtheria protein is acting on the mouse cell. So usually diphtheria is a toxin that affect the humans. Okay, so because how it affect the human, because the human cells liver cells particularly, they have receptor for this diphtheria toxin. But since the mouth cell, it does not have the diphtheria receptor, so the mouse cell will not be affected by this diphtheria toxin. But somehow, if you genetically modify it and you can express a receptor on the surface of the mouse liver cell, then the mouse liver cell will respond to diphtheria toxin and the cell will die. So these are actually used to understand the pathophysiology of a particular means substance, like that is harmful for the individual or for the human cells. And also these are used to understand the pathway, exactly how the toxin is working, so that effective therapy can be developed against them. So that is the utility of study of transgenic mouse. Then there are mouse, transgenic mouse, which are used for production of the fully human monoclonal antibodies. So monoclonal antibodies are actually the antibodies which are produced from a single clone of B cell. So they are effective against a particular pathogen or particular antigen. So in order to fight various diseases, we need monoclonal antibody that can provide us passive immunity. So different mouse models can be used for expression of the and development of the monoclonal antibody. It is similar to like purifying the protein, whatever we discussed in the bacterial case. So the gene is introduced, and after the mouse cells will the mouse will produce sufficient amount of monoclonal antibody that can be isolated, purified, and can be used for therapeutic purpose. Now transgenic livestock I already discussed. So what are some applications? Now these are used for improving the quality of the dairy cattle, like adding some nutritional component or removing some component which is harmful for consumption, etc. Then there is a development of transgenic pig, like increasing the omega-3 fatty acid content in the pig. So wherever the pork is consumed, so that is supposed to be a source of protein and source of omega-3 fatty acid. So this kind of pig, they are designed to express or to produce more amount of omega-3 fatty acid, so that when you will be consuming the pig, you will have sufficient amount of these compounds. Similarly, there is a very interesting thing, the development of a environmental friendly pig, that is EnviroPig. Okay, so usually a transgene phytase was constructed by taking the APPA from E. coli and promoter from the parotid secretory protein. This transgene was microinjected in the male pronucleus of the fertilized eggs, and EnviroPigs were created. So actually, what is the utility of this EnviroPig? Now, if you observe, so EnviroPig means it is somehow related to the environment, means they should be environmental friendly. Okay. So these transgenic pigs produce the enzyme phytase in the salivary glands, which is secreted in the saliva. The phytase gets active in the acidic environment of the stomach and digest the phytate present in the feed, so that whatever fecal matter is excreted by this pig, it has low phytate. So the phytate is actually an environmental pollutant. So because of the genetic modifications in the pig, you are having now the excreta which has low phytate. Okay. So you can go through the text, you will get to know. So this is actually the phosphorus from the pig manure run off into the water bodies and causes the algal bloom. So this is the phosphate which is released in the fecal matter of the pig, and the phosphate is actually means very harmful for the nearby water body, because it will, as it is a rich nutrition from the like nitrogen and phosphate, both if they will be present in excess amount, they can cause excess growth of the algae, which will actually destroy the means the diversity of the aquatic ecosystems. So in order to prevent that, the a pig has been designed, or the transgene has been introduced, so that the pig will be having low amount of phosphate over here. Okay. Then similarly, you can also harbor a different type of organ in the pig, and you can transfer those like liver and heart. They can be grown inside the pig, and they can be transplanted in the humans in later stage. So these are the implementation of the transgenic animals. Okay. Similarly, the transgenic fish can be also developed using certain genes of your interest, like the growth. Here you can see this is a normal salmon. So salmon is primarily used for as a source of food. Now this is a transgenic salmon. So this is simply because of presence of a growth hormone. This salmon has increased much more higher in size. That is actually fulfilling the demand. Okay. Similarly, there could be pollution indicator fishes can be designed. So pollution monitoring in the water bodies by the transgenic medaca. There is a that is a group of fish. So you can introduce a particular gene of your interest, and they can be utilized as a pollutant indicator, or they can, like we discussed in the phytoremediation, they can also be used to monitor the pollution. Then we have this glowfish, okay, the GM zebrafish, the genetically modified zebrafish, is transgenic zebrafish named that the glowfish was created for aquarium by the scientist. This fish contains a red fluorescent protein gene from the sea anemone under illumination of the ultraviolet light. The glowfish means they are mostly used for ornamentation purpose. Okay. Then next comes the genetic engineering too. Here actually we will be seeing how we can means alter the genetic expression. So we can have different types of genetic modifications, like we can have gene knockout, we can have gene knockdown, and we can have gene knock in. Okay. So knockout means removing a particular gene. So there are various methods by which you can delete a particular gene from the target sequence. So here you can see the gene knockout in yeast using the PCR based gene deletion strategy. A DNA construct with a selectable marker is transformed into the yeast, which will replace the target chromosomal gene by homologous recombination. So this is the uh, gene we are using for deleting a segment of chromosome in our target sequence. So this is the part of the 5' end of the target gene. Okay, this is similar to this part, and this is similar to the 3' part. And this middle gene is actually consisting of a antibiotic resistant gene. This is an antibiotic kanamycin. So this is resistant to antibiotic kanamycin. So now, since these two sequences are similar or they are homologous, they can undergo homologous recombination. So if homologous recombination will occur, so this part will come here, and this segment which is actually present here can be deleted. So since we are removing a part of the desired gene, this is called a gene knockout. Okay, so similarly, various other methods are there where you can actually delete the gene. So this is another method by using neomycin resistant and TK thymine kinase gene to knock out the gene. Okay, so what is done here? This is the target gene. So the target gene is disabled by the inserting gene, that is the neomycin resistant gene is inserted for the selection, and this is the disabled target gene now. Okay. Now the disabled gene is transformed to the embryonic stem cell by homologous recombination, and then you have to select whether the disrupted gene is inserted into the target animal or not, or into the target cell or not. So why we are doing this? Actually, the knockout is done to find out the function of a particular gene. So what is the role of the gene in the particular cell? So if you knock out means if you are inactivating the gene, then the cell will not have the normal function. There will be some problem, and the problem will tell you what is the function of the gene. Okay, like for example, if this is the insulin gene, and if you inactivate this gene or if you are knocking out this gene, this will mean that the cell cannot produce any more insulin, and it will be sensitized to sugar concentration. So that is the function of the knockout method. You are purposefully inactivating a gene to find out what function it is performing in a cell. So that then this construct is actually inserted into the embryonic stem cell, and when the embryonic stem cells are inserted in the mouse, they will express or means they will show the mutation or the mutant function. So how exactly we'll find out whether your means instruction the insertion has been proper or not. So this is the transgene construct or this is the vector. Okay, this is the target chromosome. So three possible things can happen. Either the gene of your interest will be inserted into the target sequence, or it will be inserted at a different place than the target sequence, that is called a non-specific integration, or it will be inserted exactly at the site where you want the gene to be inserted. So if there is no integration, your cells will be sensitive to both G418 and glancyclovir. Okay, so glancyclovir. So what is this? Now this G418 is actually means this is resistant. The neomycin gene will be resistant to this G418. Okay, and this whatever HSB TK2 is present, this gene is actually sensitive to this glancyclovir. Okay, glancyclovir. So what will happen if this transgene is not inserted? That means the neomycin is not present and this TSK22 is not present. So it will be sensitive to both. Now, if there is non-specific integration, so non-specific integration means this is the location where you want the gene to be inserted, but it has been inserted somewhere else. So since it has this particular gene, that is neomycin, it will be resistant to G418, but because of the presence of this TSK42 whatever, it will be sensitive to glancyclovir. Okay. Now, if it is exactly inserted into the site where you want it to be inserted, then this TSK22 will not be there. So this will be resistant, and because of the presence of neomycin, it will be resistant to G418. So this is your desired construct. Okay, so whatever cells will be resistant to both, that means in that cell line only your gene of interest has been introduced at an appropriate place. So these are some of the other methods like the PCR method, where the non-specific integration and the homologous recombination can be identified. If it has been, if you get a PCR band, that means the homologous recombination has taken place. Okay, if you are getting a band, that means the homologous recombination has taken place and the gene has been inserted exactly the location you wanted to. If you don't get any band, this is a non-specific integration. So you don't want this kind of integration. So these are the steps of means establishing the homozygous knockout mice lines. Can go through them. Simple steps are there. The blastocyst stage of embryo is obtained from dark color stain of mice. The embryonic stem cells are obtained from the inner cell mass. The embryonic stem cells are transfected with the transgene. Okay, that then the embryonic stem cells with integrated transgenes are selected, and these ES cells are injected into the blastocyst from the light color mice, and the blastocyst is implanted into a light color female to obtain a light dark offspring, that is the chimera. So chimera means it will be containing cells from both dark and the light mouse. So the chimera is mated with a light color mouse to obtain the offspring. The DNA from the dark color offspring is examined to determine if they contain the transgene or not. And the homozygous lines of the mouse were obtained by selfing the G1 generation, and then it was maintained. So it is like you have a mixture of different type of genotypes, but only the animal which is dark color will be actually having the homozygous transgene. Okay, and you have to select only those animals and you have to propagate them as they will be representing the homozygous or pure line genes. Then there is gene knockdown by the RNA interference. We have already discussed this, how the RNA interference works. So there is an enzyme called dicer which will be targeting and cutting the DNA sequence, and this will be actually directed by the small hairpin RNA. So it is forming a hairpin kind of structure. So in either case, the silencing RNA is there produced. So the silencing RNA is having actually a complementary sequence to that of your target DNA. So it will go and along with the dicer, it will form a RNA induced silencing complex, because of which there will be unwinding of the RNAi, and the RNAi can go and bind to the target sequence, the target mRNA sequence, where it will simply degrade the mRNA. So no mRNA means there will be no protein. So this is what is called as a knockdown. Okay, so knockdown means the gene is there, but you are blocking the expression of the gene. Similarly, here also the same thing can be done. The gene can be knocked down by use of the shRNA or small hairpin like RNA, and sometimes it is probed with a GFP to ensure that the knockdown has taken place. Means wherever the RNA will be acting and it will be knocking down the gene, you can observe the expression of the GFP, or you can observe the emission of the green fluorescent light, because this is actually coupled with the shRNA. So if you are observing a green emission, means the gene has been knocked down in that particular cell line. Then knock in means inserting a DNA, like here as we discussed, there will be elimination. Here there is insertion of the DNA. Okay, so the M13 vector, if you remember, I discussed in the early of the class that this is a vector which is used in different genetic cloning experiments. So this is the single stranded M13 recombinant, and this is the mismatch that you are deliberately inducing. So chemically synthesized oligonucleotide is there, and you are annealing it. Okay, so now if you observe this AG, here there is a mismatch. So you are creating a mutation and you are introducing the mutant DNA to find out what could be the possible effect of the gene. Okay, so this is the wild type and this is the mutant. Then you can screen by the P32 labeled oligonucleotide hybridization to find out the mutant, and you can study what kind of problems will be there. Then similarly, how the mutant is cloned, you can use different methods like multiple primer method, or you can use the single oligonucleotide method. So this is the multiple point mutation. So many mutations are there. So mutant oligonucleotide with multiple or 4 single base pair mismatches. So you already know what is the sequence of this DNA, and you are creating the mismatch mutations. Okay, so these are the mismatch point. So if they will annul, that means the mismatch is there. And similarly, you can also find the insertion mutagenesis, the mutant oligonucleotide carrying a sequence to be inserted, sandwiched between two regions with sequences complementary to the sites on either side of the target site in the template. Then you can also use deletion mutagenesis, the mutant oligonucleotide spanning the region or to be deleted, binding the two separate sites, one on either side of the target. Okay, so here you do not have means your whatever you are giving, whatever probe or primer you are using, it does not contain a particular segment. And here you are using a oligonucleotide that is having a excess segment here. So in either way, you can detect whether there has been any insertion or deletions. Okay. So this is the screening of the mutant cells. Okay. So it is almost similar to whatever we have already discussed. So you just need to isolate and you can screen by presence of different antibiotic resistance genes. Okay. So as you can see, this is the gene of your interest, and you have deliberately created a mutants here. So the transform competent cells with the mutagenesis reaction then grow overnight with the antibiotic selection mix. So this is the insert, this is the ampicillin resistant, this is the mutant form. So if this is the means wherever the mutation is not there, it will not grow because it will be sensitive to the new antibiotic. But here, as there is mutation, it will not be killed by the antibiotic. Already it will be resistant to ampicillin. In addition to the ampicillin, it will be resistant to other nucleotide as well. So this is the mutant line you are actually looking for. So this is again the PCR based mutagenesis. So PCR based mutagenesis means it will have like what you will have here. You will have PCR with primers, one and one mutant. Means you will have normal primer and the mutant primer. Then you will be removing the primers and combine the A plus B denature means A plus B denatured DNAs. Then you can extend them, and this is the site directed mutagenesis. Means this is the site where a mismatch is present. Okay, you have inserted two mutants here, first mutation and the second mutation, and you have removed the rest of the parts and you are joining them so that this part will be remaining exactly in the middle and they will be extended over here. So this is exactly, you have created a mutation at your desired site. So this is site directed mutagenesis. Then next you have the PCR based site directed mutagenesis. This method uses two different primers. One of the primer is mutagenic. After PCR, the template DNA is removed by the treatment with an enzyme called, this is a restriction endonuclease which will be removing this one. So add the PCR primer, this is the insert, this is the mismatch. So wherever the mutation is induced, okay, so you need this mutant primer. So you can use two different type of primer. There will be a normal primer, there will be a mutant primer, but this enzyme actually cannot this enzyme will be acting upon this mutant primer only. So you will be having a digestion here. Then the ligase can form the mutant insert, and this plasmid, this vector which is having the normal sequence will be simply disintegrated. Okay, so this is something similar to what we discussed here. Multiple primers you can use to identify the mutated versions where there is a single mutation, a point mutation, or there has been deletion mutations, or there are large mutations are there, or small mutations are there. These are the primers that are designed for inserting site-directed point mutations. Then comes the last part, that is the CRISPR. Okay, so this is one of the questions in your assignment also, what I went through. So there is the targeted genome editing. This is actually one of the most recent developments in the field of gene editing. So first let's understand what is CRISPR. So CRISPR stands for clustered regularly interspaced short palindromic repeats. So these are actually the means this is a defense mechanism in a bacteria. So as you know, the bacteria can be they can be infected by the bacteriophages. So the bacteriophage, as you see here, they will actually infect the bacteria and they will be inserting their genome here. So usually the bacteria has a defense system which can degrade the phage genome. Okay, so this is the CRISPR sequence. So this is the leader sequence. These are the spacer sequences with multiple repeats. So this leads to the spacer integration, and then there will be transcription and the cRNA biogenesis. Then this is the pre-cRNA, and there is an enzyme called as Cas9. Okay, so the Cas9 along with the PNS3, the PNS3 stands, RNS3, they will form a compound like this. Okay, so these compounds, they will be having complementary sequences. So whatever RNA you are observing here, they will be having complementary pairing against the phage DNA. So they will go and they will be binding complementarily to the phage DNA, and they can simply degrade the phage. So that is how the bacteria will protect itself. Okay, so the same thing is actually used, or it is exploited to target to carry out the targeted genome sequences, genome editings in humans also. So this is the enzyme called as the Cas9, CRISPR associated enzyme 9, and this is a guide RNA. Okay, the gRNA stands for the guide RNA. So these are the double stranded DNA. So the Cas9 along with the guide RNA can cut or can cleave the target DNAs. Okay. So now, since these sites are cleaved, you can actually put your gene of interest. So by two ways, the gene of interest will be integrated into the genome, that is non-homologous end joining. Means even if there is no homology, simply the DNA can go and get integrated. Or if means by some chance the donor DNA had some homology with the DNA of the bacteria, then there will be homology directed repair. So this will be the new gene of your interest will be integrated into this particular genome. So there are various applications of the CRISPR or Cas9 system in the genetic engineering. So you can just go through that. So how exactly they're used for means different kind of disease treatment, or if there is any kind of genetic defect is there, that can be repaired by the Cas9 technique. But there are certain ethical concerns related to the CRISPR Cas9 technique, so they must be dealt accordingly. And these are the questions that were in your assignment in this particular paper. So as some of you were asking about how you should be writing the things and all. So here, for example, if you look into the last question, so it tells that describe the techniques of the CRISPR or Cas9 based targeted genome editing. So for this, what you can do, here from where here the answer actually starts. So first you give a brief introduction regarding what is CRISPR. So just means mention the definition and how it was discovered in bacteria. Then you can go through the procedure, how exactly it is used or exploited for targeted genome editing, and you can put this diagram, and you can also write down the application part, so that will be sufficient for the answer. So like that, the other questions are most of the things I have already discussed, so you can just means correlate and prepare your answer accordingly. Like here, if you observe, there are fractions. Okay, so what is gene? So first you need to answer, give a brief definition of what is gene, then you explain the one gene one enzyme hypothesis along with the suitable diagram and labeling. So whenever there are fractions in the question, you must answer all the parts. Okay. So that is what I could do best from my side. I hope it will help you when you will be going through the material and all. Okay, so already the time is means already it has been over. So I'm really sorry for whatever left out. You just go through whatever part is left out, and if you find any difficulty or if you need the feel of discussing any particular topic or anything, you please request them for a class, because I have been assigned only 4 classes. Okay, so this is the best that I could do, and I hope this will help you while you are going through the topics or chapters. So if you have any other question, you can ask me related to the class. Uh, and if there is any other issue, you should talk to the means officials, okay? Because these things are not in our hand. Okay, I wish you all the best, and I hope this all of you will read thoroughly this paper, and this will means because this paper is quite important and it has a lot of application parts. So if any of you are going into the research field or anything, this is highly essential. The knowledge based on this paper is highly essential. So I have tried my best. I don't know how far I was able to convey whatever I wanted to deliver to you, but I have tried my best, and I just hope it will help you in future. And if you feel like you need to communicate, please means ask your headquarter members to means schedule any class or create any platform where we can be interactive. Okay. So thank you so much, and I'm wishing you all the best. And as per the assignment part, I have briefly means guided you. So I hope all of you will be writing the assignment in a very precise manner, and you will be getting maximum marks, scoring maximum marks in the assignment. And just one little tip, Please do not copy directly from the book. Try to modify the sentences or try to understand and write it in your own language. So that is actually highly means that actually that gives a very good impression to the examiner. Okay, so just do not copy down directly from the book. Try to modify it a little bit and try to mix it with your own language. Okay, so you need not write too much things. Write briefly, but make sure that particularly the fragmented questions, that each part of the question is answered. Okay, like for the question number 3, as I told you, first you need to write, explain what is a gene, what is the modern concept of gene. Okay, so there are 3 concepts, that is recon, cistron and mutant. So you explain that part, then you come to one gene one enzyme hypothesis. Do not directly start from one gene one enzyme hypothesis. Okay, so if you do that, then the 4 mark is gone. So you have to be very careful while answering the fragmented questions. So try to answer each fragment. Okay. So I hope you might have liked my classes, and I'm wishing you all the best for your course. If means I will be given any more classes, we will be meeting once again. Okay. So thank you so much, and for your patience, and my apologies if I have done any kind of inconvenience to you. Thank you so much. So I'll be wrapping up now. I hope there are no more questions from anyone. So let's wind up the class. Okay. So thank you once again.